Hi, everyone. So, as the title suggests, today I'll be talking to you guys about what we can know and how we can understand how the human hand evolved through studying the finger bones, or like the the actual bone structure of finger bones. So this work is mostly what I did during my PhD. Um, and now I'm moved on to a postdoc, but some of the stuff is still not quite published. I'm in the process of publishing, so it's still quite exciting and new data. And before I start off on the topic, kind of want to talk a little bit about what I'm broadly interested in and what drives my research. So before I even started my PhD, I was always interested in this transition between like the loss of arboreality and the rise of obligate bipedalism, like what the fossils at this time period looked like, what the morphological similarities and differences were between all the different species that we find between this period of 6 to 7 million years ago to about a million years ago. And because you can't study every bone in the body, I naturally got quite interested in the hand. Particularly, I got interested in how a hand of a hominin ancestor somewhat looked like probably what a great ape hand looked like. So really long fingers, really short thumbs, a hand mostly used for, locomotion, grasping, to a hand that somewhat looks like what we modern humans have now, where we have really long tongues relative to our fingers, and we're capable of quite precise and quite complex manipulation. And really forceful manipulation as well, which because of the different hand proportions and a variety of other morphological features of the hand in great apes, that's not really possible. So how do we actually reconstruct human hand evolution? How do we kind of know what the transitionary steps were between a hand that was primarily used for locomotion to one that's now mainly is used for manipulation? And as you guys can probably guess, we do that by studying fossil hominin hand remains. In this diagram here, you can see a couple of the key species from which we have an abundant amount of hand remains, because a lot of them end up being quite fragmentary. And some of these species I'll be speaking about later. So when we get the fossil hand remains, what we really look at is the external morphology of the bones. So what they look like in shape, just their overall shape, but also different aspects of the shapes of something like the articular surfaces or the muscle attachment sites. And we can also look at the internal morphology, which is the internal bone structure, because it adapts throughout life, and it can tell a lot about what an individual was doing throughout their lifetime, which I'll talk more about today and is the main focus of my talk. So in order to actually understand what kind of like the shape and the bone structure of fossil hominin actually means and what the behavioral implications could be, we then look at great apes as a comparative sample, because they're our closest living relatives, and we actually know a lot about their morphology and how it relates to their behavior, which so we can link these different bony features to what they're actually doing and what they're behaving like, and that tells us kind of like the purpose of each I guess distinguishing morphology. So if in the fossils you see, a mix of like ape-like or human-like signals, you can potentially weed out what the purposes of that morphology is. And that's how you can reconstruct fossil behaviors. And obviously that's just not in the hand. Most paleoanthropologists that do functional morphology or try to reconstruct fossil hominin behaviors across any bone of the skeletons kind of do that, where they compare any fossil remains to the great apes. And kind of furthering on to why I'm really interested in the hand, it's because we can truly address some really fundamental questions about human evolution by looking at the hand. And the first one being is like, when did the locomotor use of the hand cease? Because traditionally the thought process is that something led to the development of obligate bipedalism, and that inherently has to happen after we stopped, being in the trees, using the trees to move around. And that's still not completely used. So by studying the hand in more detail, you can kind of get a closer answer to when that transition between being in the trees to being on the ground constantly and consistently actually occurred. And secondly, when and how did tool use evolve? So tool use is obviously linked to a lot of, traditionally has been linked to a lot of developments of our brain, access to like meat resources, and just tool use had opened up for our fossil ancestors a whole different part of the landscape that they could exploit after gaining access to tools. And traditionally, there's this kind of like dichotomous view that either the hand was being used for locomotion or manipulation, that the two can't kind of exist at the same time together, because surely a hand that's adapted to be used in locomotion could not perform as effectively at making or using stone tools. But as we know, evolution doesn't work that way. It's not that one day we were in the trees and the next day we were on the ground using tools, not using our hands for locomotion at all. So studying fossil hominin hands can shed a light on exactly when that transition occurred, because it was a transition where it is likely that we were using the hands for both locomotion and tool use, and at the same time we were walking on the ground, maybe definitely not all in an obligate manner, but those things were all occurring together. So what kind of that transition is that led to manipulation being the primary use of the hand and obligate bipedalism. So, and if you guys are still wondering why the hand is so interesting and why I want to study it beyond these questions, we can also like potentially answer these questions using the other bones of the upper limb. So the hand might not be, a priority, or might not seem as important, because obviously the shoulder, the upper arm bone, all those things are quite important in climbing as well. But the reality is that across all primates, the first point of contact with any substrate or object during any sort of like manual behavior, so whether that be locomotion or manipulation, is the hand. The hand touches down first, it grabs onto a tool first. So the hand is actively being impacted by those direct forces, and the bones of the hand are then being shaped by those direct forces. So within the hand, I'm personally really interested in the phalanges, because as a whole, the hand touches down first and makes initial contact during those locomotor or manipulatory behaviors. However, phalanges are the ones that touch down first. So if you picture kind of a chimpanzee or gorilla knuckle walking, it's the phalanges that are touching the ground. Or if you're an arboreal primate in the trees, you're wrapping your fingers around a branch first. You're a hominin using stone tools, you're holding it, and the fingers are providing kind of like that contact, that initial contact phase. But when we look at studies of the hand, studies on the phalanges are comparatively lacking when you compare it to the wrist or the metacarpals. But that being said, there's certain aspects of phalangeal external morphology that have been studied. And the most studied aspect of the phalanges is actually phalangeal curvature. So it's this, just essentially this angle of the phalanges. And it's been shown that the greater the curvature, the greater the degree of arboreality in a primate. So there's a lot of variation across all the primates, and it is linked to how often they spend time in trees. And you can see that in the great apes and across fossil hominins as well. So orangutans being kind of like the most curved, humans being quite flat, and then gorilla and chimpanzees are somewhat in the middle, with chimpanzees having greater curvature because they're more arboreal than gorillas. Another aspect of the external morphology of phalange that has been studied are the flexure sheath ridges, which are these ridges on the radial and ulnar aspect of the phalanges. And essentially, they just protrude out from the palmar surface of the hand, so the front of the hand, and they're prominent. So how far they kind of come off the surface of the bone has been linked to arboreality and has been considered advantageous for being in trees or suspending and climbing behaviors. So coupling this varying external morphology with the study of bone structure, which for those of you who don't know, bone structure contains cortical bone and trabecular bone, cortical bone being like this outer bony shell, and trabecular bone being this spongy porous bone, typically on the ends of long bones, the epiphyses, or all within the short bones. Both trabecular and cortical bone have been shown to adapt to loads placed on it throughout life, a concept which is known as bone functional adaptation, which essentially states that areas that are highly loaded in a skeleton, so areas that are undergoing high loading consistently and constantly experience bone deposition, so there's more bone there, while areas that aren't being as stressed, aren't being as used consistently, undergo bone loss. And an example to kind of show that is this. This is a humerus of the same individual. It's a left and a right. It's of a professional baseball player who uses one of its arms to pitch and the other one he doesn't. And when you look at the cortical bone at cross section, you can really see the difference in just one individual. So throughout this person's lifetime, however long they've been playing sport, they've only consistently been using one arm for throwing, and that's over their lifetime has led to a massive increase in their bone density. So as you can see, studying the internal structure could really tell us what an individual was doing throughout their lifetime and what their habitual behaviors was. So for fossil hominin, it really does provide us with a great framework from which we can kind of get to what they were doing regularly. And for the hand, our understanding of the hominin hand and hominin hand evolution has been relatively recently been greatly expanded through a lot of discoveries, particularly two incredible discoveries of nearly complete and associated hands, which one of them is Homo naledi and the other is Australopithecus sediba, which are both hominins from South Africa. And we have a lot of other fossil remains as hominin hand remains of different species. And some of the other more complete ones are Homo floresiensis, even though it's multiple different individuals. We do have kind of somewhat of a composite hand. And then another one is Homo habilis, or known as the 087 hand, which some of you might know is quite a key species in human evolution, as it was dubbed one of the first like Homo species, and also is found in close relation to Oldowan tools. So it's supposed to be the first indicator of, when we're actually using tools and whether or not this specimen can show that is what we test here. And when studies of the external morphology of these fossil hominins have been done, they kind of give indications of both manipulation and locomotion signal in the hand. And when we look at the internal structure of these bones, so mostly the work's been done on the wrist and the metacarpals. The results have shown, so people have looked at like trabecular structure and muscle enthesis, so muscle attachment sites, and it's been shown that generally the manipulation signal is held within the radial side of the hand, so essentially the thumb and maybe the second digit or the second finger, the index finger, and the locomotor signal tends to be in all the other aspects of the hand. So the external, obviously these are giving quite conflicting signals, and it's not a clear kind kind of clear-cut answer of which species were manipulating more, which species were locomoting more. So our next step is then to look at the fingers, as they can maybe shed more light and maybe provide some clarity to some of these responses as well, because obviously there are a lot more. Yeah, as I said earlier, they make that initial contact first. So now I'll actually get into kind of what I've done, and here is what my sample consists of. So we are looking at species all the way from nearly 4.2 million years ago with Australopithecus afarensis all the way to Homo floresiensis at 100 to 60,000 years ago. And we did this by using micro-CD data of phalanges. And for the afarensis material, we have just one isolated phalanx. For Africanus, we have a couple of isolated remains. And for Australopithecus sediba, we have the full associated hand with the second intermediate phalanx missing due to just it was broken. A lot of it was broken and crossed, so it wasn't useful for analysis. And for Homo habilis, we have all the preserved phalanges, Hominoleti, the complete set of phalanges. And then Homo floresiensis is a couple of phalanges from different individuals that none of them are confidently associated. And before I do start, I want to address the notable lack of Australopithecus afarensis and Africanus phalanges. There are a lot more fossil remains that do exist, but sometimes the internal structure is quite hard to study because of the preservation of these fossils, which we can see here. So this is kind of what the micro CT scans of the phalanges look like, and this is just one phalanx from each individual. And as you can see here, the preservation varies by quite a bit. So we can sometimes get really lucky with fossils and get something that is so clean, which, just it has a little bit of inclusions, but nothing too hard to clean out. Or you could get something like Naledi and Sediba, where the bone, even the cortical bone, even if the bone is clean on the inside, due to like mineral deposits, it changes the density values within the bone. So it's quite hard to get a proper segmentation out or make all aspects of the bone quite clear. And then you could get really unlucky and get something like afarensis, where you can barely distinguish the matrix from the bone itself, which is why for afarensis we only have one, because this is the only individual where we could kind of really get the cortex out of the inner structure. So how do we actually study the cortical bone through this micro CT data? After we clean and segment the bones, we create inner and outer surfaces for each phalanx. So the outer surface being this cortical shell, and the inner surface will include everything that's in it. So we don't include this trabecular spongy bone, and we put that all in an inner surface. And this is what it ends up looking like. The red is the inner surface and the gray is the outer shell. And we're really just interested in the phalangeal shaft, and that's because of multiple reasons, particularly because at the epiphyses, it's complicated to reconstruct internal bone structure, because you also obviously have the trabecular bone playing a role as well. So you can't just, I think, isolate the cortical bone. So while the shaft is just the cortex, and there's been a lot of studies that kind of reinstate the importance of looking at cortical bone across a long bone shaft. So after we get the surfaces, we define the shaft by cutting the articular ends off. And then we use an R package called Morphomap. And what Morphomap does is it places cross sections throughout the entire length of the shaft, and you can define where you want those surfaces. We did them at 1% increments throughout the shaft. And at each of those cross sections, you can place landmarks on the inner and outer surface to that really capture the shape of the cross section and of the bone as a whole. And then once those landmarks are placed, from the center of the bone, you shoot out rays outwards towards the outer and inner surface. And cortical thickness is calculated at each point as the length of the segment between the outer and inner shell of each bone. And using this methodology before we applied it to fossil hominins, as I said earlier, we need to look at the great apes, and that was my kind of first step into doing this analysis before we got to the fossils. And when you do that, you end up with something like these color maps. So we're going to see a lot of color maps throughout this talk today, and the important thing to remember is that the areas where you see red, that means the bone is thickest there, and the areas where you see blue, that means the bone is thin there. So for orangutans, generally see this thickness on the palmar surface of the hand. So again, the front of the hand, and particularly on these ridges that I mentioned earlier, the flexure sheath ridges, but just at their peaks, so not across their entire length. And that kind of is quite consistent with what we know about orangutan locomotion, which is just this power grasping posture where they grasp around really thin branches or really thin substrates and support their body weight below the hand. And for the African apes, we again see a similar pattern of having thick cortical bone on the flexure sheath ridges, but in a varying manner for both gorilla and chimpanzees, which is reflecting their different behaviors. Because with knuckle walking, your hand is in a flexed position and is being loaded palmarly, but with the body weight on top of the fingers this time compared to orangutans, which have the body weight below the fingers. But also, gorilla and chimpanzees also have different levels of arboreal behaviors in them as well, which is why you could see why gorilla has this more of a patched cortical thickness, while chimpanzees are quite consistent throughout the entire ridge in the front. And generally, these great apes show this pattern of thickness, like I said, because of their different locomotion. So here, you can see this is what an orangutan kind of finger would look like around a branch during the locomotion, and the depiction on the bottom is what kind of a chimpanzee or a gorilla hand or finger will look like during locomotion. But the key thing to point out is that they're both in a flexed position, which is why you kind of see this thickness on the palmar surface. Because as we mentioned earlier, the prominence of the flexure sheath ridges is to dissipate any loading from arboreal behaviors. And in arboreal behaviors, the primary action that's happening is that the fingers are being flexed and they're being loaded on the palmar section where the musculature is being extended, and they really have to have a good grip to stabilize the hand or the finger joints and really hold on. And while that looks very different in knuckle walking, the finger is still flexed. If you look at kind of like the depiction on top, where the bones are kind of wrapped around what's supposed to be the substrate. Here, the bones will also need to be the ligaments and the joints be loaded here as well, because they need to then stabilize the finger joints so that when chimpanzees and gorillas are supporting the entire body weight on top of the finger essentially doesn't buckle and it's quite stable throughout the bout of locomotion. So it's basically flexion. Strong, powerful flexion results in this palmar pattern of cortical thickness across the non-human great apes. And then when we look at humans really do display this unique dorsal pattern of cortical thickness. And this dorsal cortical pattern of thickness has likely to do with how loads are being dissipated across the fingers when the fingers are relatively straight, but also they're not being loaded as heavily. They're not being used to support any sort of body weight in any way. So quite a clear distinguishing pattern between a hand that's being used for locomotion versus manipulation. And we see that in statistical analysis as well. So we take this data and we put that into PCAs. And this is kind of what the data looks like that we put in. These are just unrolled versions of the 3D maps. And you can see that humans are statistically different from the great apes because of this dorsal thickness, while the great apes on one end are determined by this palmar thickness, with the non-human great apes also showing quite decent separation due to their different locomotory modes. So now that we have a kind of a comparative context set up, we'll get into the fossils. There's going to be a lot of color maps being shown for each of these phalanges that you see here. So what I would like for you guys to remember, so it doesn't get as overwhelming, is that anytime you see a palmar signal of cortical thickness, associate that with locomotion or the non-human great apes. And anytime there's a dorsal signal, you can associate that with being more similar to humans. Right, so let's get started with the earliest fossil that we have with the single phalanx of afarensis. So here you can see, even though it's not as strong, one second, I have the window. Yeah, so the cortical thickness is thickest kind of on the palmar surface, even though there's just a little bit of it just towards the end of the phalanx. The cortex is thickest there, and when we plot that in the PCA, it does come out closer to pan. And when we look at Africanus, again, we see quite a similar pattern with cortical thickness being located on the palmar surfaces in both the proximal and intermediate phalanges. And we also have this proximal phalanx, so the phalanx of the thumb, that also shows a great ape-like pattern. And when we do PCA analysis, again, you can see Africanus falls quite close to pan. And for the intermediate phalanges, it is somewhat in the middle. And for the pollical phalanx, quite nicely into the great ape distribution. And now we'll move on to our first associated hand with Australopithecus sediba. It's quite important that we have a full kind of set of associated phalanx, because we can really then map out what this individual was likely doing, but also why, like the unique mix of morphologies that they have and what that tells us about their locomotor mode across the entire hand. And here you do see how the mix of more ape-like and human-like morphology shows up, because across the phalanges, you do see this palmar pattern of cortical thickness. However, when you look at the dorsal side on the fifth digit, you do end up seeing a little bit of the dorsal thickness. It's not as strong as the actual human signal that we see, but you can see at the tops of the fifth digit, so the pinky finger, you see a slight bit of dorsal thickness coming in, which is why for the proximal phalanges, sediba ends up being quite close to the pan distribution. However, for the intermediate phalanges, particularly in up in digit 5, you can see it falls kind of in the middle. But for the pollical phalanx, so the thumb, it is sitting right well in the middle of the great ape distribution. The next fossil we have, like I mentioned earlier, is quite important, because when Homo habilis was found, it was, like I said, found in close proximity to older one tools, and it was quickly dubbed the handyman, alluding to its manipulative abilities. But the robust external morphology and palmar cortical thickness of the phalanges is kind of at odds with this interpretation. So it's not just that the palmar cortical thickness is giving us this ape-like signal, but these phalanges are quite broad and robust. You can see it in their muscle markings here. And it's really surprising, particularly because this individual is also a juvenile, nearing adulthood, but estimated to be still a subadult. So to have this really strong muscle markings as a subadult and have this really robust look that we haven't, when this was discovered, haven't yet observed in fossil hominins that were known or that could potentially use tools, is quite unique and is kind of at odds with it being found in such close proximity to stone tools. So for the proximal phalanx, it falls quite close to the pan distribution, and just at the edge of the pan distribution actually, and the intermediate phalanges as well. So again, indicating a more being more close to the non-human great apes rather than a human-like signal. Right, moving on to Homo naledi. Homo naledi is also another one of the complete fossils that we have, and here we really do see a mixture of patterns more strongly than any of the other fossil hominins, where it's quite the palmar signal is quite clear and apparent in the intermediate phalanges and in the proximal phalanges, but the proximal phalanges also show this really strong dorsal pattern, which is something that we haven't observed before, in any of the extant sample or in any of the fossil samples. And when we plot that in PCAs, surprisingly, Naledi falls close to humans in both the proximal and intermediates, despite its pattern and its thumb as well. So it's indicating this really strong mixture of human-like and non-human-like pattern thus far in the record. And finally, we have Homo floresiensis, which, looking at just the proximal phalanges, similar to afarensis in a sense where the thickness is really just towards the distal end. Homo floresiensis kind of falls close to the great ape pattern. And when we look at the intermediate phalanges, they really show a completely different pattern to anything that we see in the extants at all, or the fossil hominins, but in a very strange way due to this like patched manner across the entire phalanx, which is something we haven't quite made sense of yet. But we think that could be because Floresiensis just looks very strange from like a bone structure perspective. And if you know anything about kind of like the environment of where Flores was found, which is a tiny island in Indonesia, is that island animals do have weird mixes of morphology, which could explain why kind of Flores looks a bit different to what we would expect. And it kind of falls out differently as well, falling out between humans and non-human great apes in its range. But the thumb phalanx does fall in quite nicely with the great apes, like the other proximal phalanges. Right. That was a lot of information. So I'll slowly go through, rapid fire go through kind of what we covered. So the Australopithecus and Homo habilis kind of show this ape-like pattern of palmar cortical thickness across both their proximal and intermediate phalanges. Homo naledi has this mix of human-like and a great ape-like pattern in the proximal phalanges, but just a great ape-like pattern in the intermediate phalanges. And Homo floresiensis is also showing great ape-like pattern in the proximal phalanges, but the intermediate phalanges are quite unique, and they just have a really patched pattern of palmar and dorsal cortical thickness. I think I'm running out of time, so I'm going to just skip that slide. So based on what we know about the cortical bone distribution pattern, but also kind of what studies that have been done on other aspects of the hand of these fossil species, we can say that the cortical bone morphology of Australopithecus afarensis, africanus, and Homo habilis kind of is more consistent with a hand that was used for locomotion. And that is not to say that they weren't using tools, they weren't capable of that, but that the consistency and the frequency at which they were using their hands for locomotion versus manipulation was probably leaning more towards locomotion in their habitual lifestyle. And for Sriba, we have this phalangeal cortical signal that says primarily used for locomotion, but then the fifth digit comes in with a slight bit of dorsal thickness. So indicating both use of the hand for arboreal locomotion and human-like manipulation, which has kind of been seen before when the internal structure of the metacarpals was looked at as well, because they found a climbing signal in the metacarpals 2 to 5. So these, but a really human-like manipulation signal in the thumb. So maybe different signals from the fifth digit compared to the thumb, but overall the hand is showing quite a strong signal of having the ability to consistently perform well in both locomotion and manipulation behaviors. While Naledi truly does indicate differing loading of both proximal and intermediate phalanges, and really standing out uniquely in the fossil hominin record. And few people have suggested that this could be an indication of rock climbing, particularly because the environment would have supported the ability to climb rocks, because it was a cave-like environment, and that could have been possible, particularly when rock climbing grips are looked at. The distal end of the finger is bent a lot more in rock climbing compared to the proximal end, which could explain the really the great ape-like pattern in the intermediate phalanges compared to the proximal phalange that has a more human-like pattern, because they're not being as flexed as much, which you can see in this diagram here. And then finally, Homo floresiensis has completely, just distinct behaviors and dexterity, particularly because when you look at the internal structures, these are just some cross sections here, you can see how different the bone looks like compared to any extant analog. And even other fossils don't really look this porous. So that would have had an impact on their biomechanics and how they function. So what does this what does it mean for human evolution? I think all these results put together, so not just my results from the cortical bone, but also everything that we know about other hand bones, is that these results represent a transition in manual behaviors, and a transition that's not linear in any way. Fossil hominins kind of have this unique mix of mosaic external and internal morphology that would have allowed for a diversity of behaviors. And this concept really does get lost in this dichotomy of a hand either being used for manipulation and locomotion. But in reality, it is much more likely and probable that fossil hominins were using their hands for both locomotion and manipulation, but just in varying frequencies, and that the locomotory use of the hand likely persisted longer in the fossil record than presently thought, or even, the development and use of stone tools. Yeah, that is it for me. Oh, yeah. Yeah. And I'd like to thank everyone with my funding agencies. Yeah. Any questions?
Thank you very much, Paul. A very interesting talk. Quite curious. If anyone has any questions, please put them in the chat or just raise your hand, and we'll get to you. I've got one of my own just whilst we wait. Back on your colour maps, it's quite curious to see that the Homo sapiens had quite a high density of thickness, even compared to some of the great apes. Now, I assume the data is sort of relative to each species, or is the Homo sapiens?
Yes, sorry, yeah, I didn't clarify about that. Sorry, yeah.
That
So
makes
each
sense.
each of the color map you saw, it wasn't necessarily that if the humans had such a big splotch of red, that meant they're much more thicker than the great apes. It was just, yeah, it's individual to each bone. So it was more about the pattern than the actual, I guess, level of thickness. Yeah.
Very good. And we've got a raised hand from Curate. Do you want to just
You're still
unmute
muted,
yourself?
yeah.
Hi, good morning from the
Hi, good
United.
morning.
I just was wondering, that was just a remarkable lecture. Thank you very much. There's been some speculation, well, not speculation, there's new finds of Paranthropus with tools. I don't know if there were any hand bones present with the fossil finds, but would you speculate on what Paranthropus might have if it was indeed a tool user?
Yeah, so I'm really trying to get my hands on those fossils. So yeah, I've heard they do have hand bones, they do have phalanges as well. But I've looked at a few just 3 phalanges from swart crowns, so we don't know actually if they're Paranthropus or not, or they're early Homo. But I have looked at them, and they look weird. They look really different, particularly because they're really robust in their morphology as well. So they don't kind of, for what we would expect of based on its external morphology, they aren't really giving a great ape-like signal or a strong manipulation signal either. It's falling right in the middle, but also an important thing to acknowledge with these fossils is, which I didn't really touch upon, is that while the cortical bone gives us these signals, right, that we can be like, oh, locomotion or manipulation, but the external shape of all these bones is so different from one another that we really need to look at kind of like the relationship between the external shape and the cortical bone to see how a thick, kind of like what a thick palmar cortex in a bone that's really curved or really robust looks like. So I think even if at least the bones that I've looked at from swart crowns, if they're giving us a different signal, that doesn't mean that manipulation wasn't a big part, they couldn't participate in it, but just that manipulation in a bone that is essentially just so chunky and big looks very different in a bone that's really what's the word's escaping me, but a bone that's just not as robust and is quite small, if that makes sense.
Yeah. Okay. Thank you. Thank you.
And I'll call on Walter now. You'd like to unmute yourself as well, Walter, please.
Pardon me? Can you hear me now?
Yes.
Okay, great. First, I want to congratulate you. That's excellent work and a very fascinating presentation. And I have a particular interest in human anatomy. And I want to wish you all the best in your career. But to get to my questions, hand bones are notoriously difficult to find as they're often dispersed. They're very small. So how many bones of the hands do you need to make a reasonable reconstruction about a particular hand's function? And my next question is, have there been fossil skulls found nearby to correlate brain volume, especially the posterior frontal and parietal size with brain anatomy? I'm interested in not knowing the individual hand anatomy, but you need a good brain to make that hand use, right? I mean, the brain sends out the messages for the actions. So the posterior frontal will be the motor cortex, and the parietal area, the proximal parietal area, will be the sensory cortex. So has anyone traced the skull size and tried to correlate it with hand anatomy to see if there have been any changes, or there is a correlation, or was there a rapid jump or a slow, steady jump from one hominid species to another? Did I make myself clear?
Yes, yeah. So for the first question, in terms of having like the amount of hand bones, so there's certain bones in the hands that are quite, I think, important. I can tell you a lot, just even if you just have one. So certain aspects of the radial wrist, so maybe the bones that underlie the thumb, the wrist bones that underlie the thumb, and maybe the second and third metacarpal would be quite important because they're really important for specific joint functions for all these movements. So whether that be manipulation or locomotion. So if you look at the trabecular bone structure of those bones, that could be just quite informative alone. And then generally, I would say the third digit is quite important anyways, all across the bone. So whether you get the third metacarpal, you get third proximal intermediate phalanges, because that's just the midline of the hand, and the third digit plays an important role in manipulation as well, but also a lot of the forces for locomotion kind of like are highest in the third digit. So if we were to kind of if you were to get lucky in the fossil record, if you got a couple of the radial wrist bones and the thumb metacarpal and the third digit, it would be quite a good shout at reconstructing locomotion or any sort of kind of manual behaviors. And as for your second question, so I know there's a team in Tübingen right now that's working on doing EMG studies and stone tool making and using, I think. So, and they're kind of like the first people that I know of in terms of doing something with the brain and how it relates to stone tool use. But for the fossils, based in my sample, what's really unique about, I think, Naledi and Sadiva is that they're really tiny hominins. Overall, they're really tiny. And obviously, Flores is just a beast in itself in terms of its body proportions are really weird. Because their hands are, in a sense, normal adult size. If you reconstruct like hand proportions and hand lengths, but they were a really tiny individual. They were an adult individual, absolutely fine, no medical anomaly, like nothing pathological, sorry. But their brain size is just not, obviously they have a really small brain case. So I don't think there's been any studies looking or like correlating those two things, but there is a wide range of hominin, hominin brain sizes to the hands. There hasn't been that correlation made, I think, at least.
I think it might be something worth looking into in the future.
Yeah. I think people who are more interested in kind of like the experimental application of stone tool use and stone tool making are working on it. A lot of experimental stuff in that aspect of the field that happens, particularly in experimental archaeology, I think.
All right, thank you.
Very good. Thanks, Walter. We've also got a question in the chat from Linda. Linda asks, if you hypothesize that manipulation began with botanic materials rather than with stone, this would prioritize fine motor control over force. Are there ways that this behavioral change would be visible in the bone?
I think so for botanic materials, so we do see in the wild, chimps and gorillas and other animals use these kind of botanic materials as tools, but it's really not consistent or regular enough. And the motor control to really kind of use those really fine materials would be really, I think, difficult based on what we know about kind of fossil hominin hand proportions. What the another ways that this behavior change would be reasonable. So the thing about identifying kind of behavioral signals in bone would be that we can predict habitual behaviors. We can predict the behaviors that would be likely occurring throughout life or occurring with such an intensity that they have the ability to remodel bone. But we'll never be able to reconstruct the entire extent of an individual's behavior. And you know what? A really good example of that is, so I don't know if any of you guys have seen this picture of mountain goats that climb trees. So they there's a picture of like 10, 15 mountain goats just on top of like tree branches. And they also climb like really a steep mountain face and they kind of just stay on them, like kind of stand parallel to them like that. And if we were to find like a fossil mountain goat, we would never look at its bones and be like, oh, that could climb trees. So I think we'll never be able to, fully reconstruct those behaviors that aren't inducing enough force to really restructure bone. So I think a lot of our interpretations will never capture the full extent of what a fossil individual was capable of doing. So if that answers your question.
Brilliant. Thanks, Linda. I've written another one of my own, which I think you might have just answered, but I'll go for it anyway. I'm glad you brought up the hominid rock climbing hypothesis because I think it's really quite a good idea. I have seen the paleo-environmental research. It's quite limited, but a lot of the time they suggest there's not much trees there. Maybe there was, maybe there wasn't. Maybe hominid was a rock climber. My question is, do you think using your research methods, you could tell the difference between a rock climbing phalange and a tree climbing phalange? And you could use comparative data from baboons that are more rock climbers than tree climbers.
Yeah, I think I would like to, but no, I think a lot more. I think more research is needed on the experimental aspects of rock climbing. I know that's being done currently, but I think we don't exactly know how that impacts bone or kind of what that really looks like from maybe a skeletal perspective, from a joint perspective, what exactly is being loaded. And I think to even do that regardless, because there's so many joints involved in rock climbing. And while in regular climbing as well, the one good thing about like arboreal, like primate climbing is the fingers are in quite a similar position no matter kind of which bone you look at. They're all flexed around a substrate. If you're climbing, you're holding on, your metacarpals are flexed, your fingers are flexed. But rock climbing, you could have certain aspects of the finger that are like highly bent and the rest is straight, or like different grips that you would need, I think, a more complete full digit to reconstruct that, I think. But yeah. And then
Right.
maybe a more sorry.
No, go ahead. Go ahead.
I was going to say maybe and a more, I think, integrated approach of kind of not just how the bone structure is changing, but also how the shape is changing with that as well, I think. It's probably
Yeah.
will be needed to do something so specific, such as to reconstruct such a specific grip.
Yeah, it'd be a tricky one. And do you have the distal phalanges for hominility as well?
I believe, do we? I think there's one or two, I believe. I don't think all of them are preserved. Yeah.
Would have thought so. I have a cast here actually, and it seems to have them all.
Yeah.
Maybe we do. That might be something to look
Yeah.
into. But anyway, thank you very much for answering my question, and thank you for coming on to talk today. Very
Well,
interesting.
thanks for having me.
And thank you to everybody who came to watch. Have a good rest of your day. Take care.
Bye, everyone.